Can somebody confirm you can hear me?
We can hear you.
Thank you. All right, somebody had a question. What was your question?
My question was for the unknown project for the gram stain. Do we um look at the arrangement of the gram in the gram stain as well, or just the shape and the color?
For the gram stain, it tells you that you cannot determine the cell arrangement. If you want the cell arrangement, you have to do the cell arrangement test.
Okay. Thank you.
All right. Okay, let me share my screen here. My computer is going to be a little slow because we're actually at 93% of the lecture being made. That's a little further than I thought it was. Let me see. There we go. Share my screen here. Actually, before I share my screen, let me talk about the grading. Um, I have graded some of the labs. Boy, it doesn't look like I have that open anymore. I got the last lab, I think it was 8 graded. And that one I didn't get graded, I think, until early this morning. So you will have 1 week from this Saturday to turn it in, meaning it won't be due this Saturday. It will be due next Saturday. Any question about any of that? I think it was lab 5 that I also got graded. And that one I got graded earlier this week. And let me make sure it's lab 5, but I think it's lab 5. And this one, if you want to have it regraded again, you have to have it resubmitted by this Saturday. Any questions about the grading? I got some lecture assignments graded, and I think some extra credit assignments graded. Um, I still have lab 6, I think, to grade. Grade the first time. And it looks like for grading the second time, I stated that earlier, I didn't remember. It looks like lab 1 is graded 2 times. That's the only lab that's been graded 2 times. All the other labs, I will get them graded a second time, but the first grading of the labs is a priority. And so my priority will be getting lab 6 graded and lab 9 graded. Once those are done, I'll then go back and start grading the labs a second time. And by the way, I was correct. I got lab 5 regraded. And if you want it, you didn't get a 100 out of it, 100% out of it. You'll need to resubmit it by this Saturday, 11:59 p.m. this Saturday. But lab 8 is graded, and very few people got a 5 out of 5 on that lab. Well, that can't be right. Somebody I got 5.6, and that can't be right. I must have done something wrong. Let me look on that. You're not seeing this, right? Yeah, that should be 4.6. So lab 8 is graded, and you'll need to submit it not by this Saturday, but a week from this Saturday. Any questions about any of the grading? I got some of the extra credit studies graded. I still haven't got the extra credit study sheet graded. I'll need to work on that. And I got extra credit study 6 graded, maybe 5 graded as well. And then some of the lecture worksheets I got graded. I need to get lecture 8A graded. That was last week's. If I remember right, I need to get some people graded on the last quiz, not the quiz you just took, which would be quiz 4, but quiz 3. Some students did not send me a complete environmental scan, and so I'll need to spend time viewing their entire video to see if they ever take their eyes off the monitor. If they do take their eyes off the monitor, I will assume they're reading something to cheat, and they will get penalized for that. But with the quizzes, I've decided it takes too long to do that, and that's why I finish grading the quiz, and then I'll go back and grading the students who didn't show me their complete environment around their computer. Any questions about any of the grading? All right, let's close out of here. Now I'll share my screen. So today is November 13th, and we should be discussing Lab 9, identification of gram-negative organisms. Any questions about what we're doing? I think I can close this down now. Lab 9, identification of gram-negative bacilli. You will need to finish this lab module, watch the videos, complete the lab worksheet, and complete the case studies for this lab. There's a little bit of reading for this lab, only a page on biochemical tests, and then a few pages on Chapter 10, methods of classifying and identifying microorganisms. The learning objectives are: 1, be able to interpret the results for the following biochemical tests. The IMVIC reactions. IMVIC is an acronym for the indole test, the methyl red test, the Voges Perskure test, and the citrate test. All of these tests are important, and especially family Enterobacteriaceae. And also know about the carbohydrate fermentation test, the motility test, and the hydrogen sulfide production test. In addition to these, you should also be able to explain and interpret the TSI or triple sugar iron test, the SIMS test, which is sulfide indole motility test, and the growth on McConkey's agar MAC plates or hectoenteric agar plates or HE plates. All of these are multiple tests run at the same time. The triple sugar iron, the SIMS test is running 3 tests at one time, the sulfide indole motility test. McConkey's agar is looking for cells that can grow on McConkey's, meaning gram negatives, as well as looking for the fermentation of lactose. So all of these are running more than one test at one time. And then 3, use biochemical analysis to solve case studies involving gram-negative bacterial infections. And I meant to say for the other lab that in the lab, I noticed in lab 8, in grading it, that a lot of people got some of the case studies wrong. And I should have gone through and talked about how you do a case study. You guys can ask me when we get to the case studies about that. All right. In this lab, we're going to talk about the family Enterobacteriaceae. Members of this family are all small, gram-negative, non-spore-forming bacilli. They're all facultative anaerobes. They're all glucose-fermenting and oxidase-negative organisms. There are 53 genera in this family, so this is one of the largest, if not the largest family of bacteria. 24 of these genera have been known to cause human infections, including these common genera which cause human infections. Most of these genera are normal microbiota of the intestinal tract, aka we call them coliforms because you can find them in the colon and in the intestines. They are all opportunistic pathogens in that they can cause urinary tract infections, wound infections, pneumonia, and septicemia. Common genera responsible for causing opportunistic infections include Escherichia, like E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. Several of the genera in family Enterobacteriaceae are associated with gastroenteritis, including the organisms of Salmonella, Shigella, and then E. coli serovar 0157H7. Normal E. coli is in our guts and does not cause us any problems. In fact, it actually helps us digest our food. However, E. coli 0157H7 is a human pathogen, and somebody who gets it is likely to get gastroenteritis. Any question about any of that? 0157H7 are different antigens, and this strain of E. coli is a human pathogen. There are other strains, but this is one of the worst ones. All right, any questions about family Enterobacteriaceae? We're also going to talk about the family Pseudomonaceae, particularly Pseudomonas aeruginosa. This family is similar to family Enterobacteriaceae in that they are gram-negative bacilli. However, all members of the Pseudomonaceae family are non-glucose fermenting, and they're all oxidase positive bacteria, as well as the fact that they're all aerobes, and that differs from the family Enterobacteriaceae because they're all facultative anaerobes, they can all ferment glucose, and they're all oxidase negative. Pseudomonas aeruginosa is an opportunistic pathogen causing infections, particularly in immunosuppressed individuals, burn patients, and trauma patients. Fortunately, Pseudomonas aeruginosa cannot easily get through intact human skin or intact mucous membranes, so it rarely causes an infection in a healthy, normal individual. However, burn patients and trauma patients have their skin compromised or the mucous membranes compromised, and it can frequently infect those patients, as well as immunosuppressed individuals. Pseudomonas aeruginosa is also implicated in healthcare acquired infections. We've discussed these before, more as nosocomial infections, and this is an important nosocomial infective agent. Of course, remember, the number one most common nosocomial infective agent in the United States, at least today, is E. coli. All right, it's important for microbiologists to be able to identify and perform antibiotic susceptibility testing on gram-negative bacilli that are cultivated from clinical specimens. In this lab, we're going to talk about some common biochemical tests used to identify gram-negative bacilli. And here are some tests here. We're not going to talk about that many, but here's a bunch of tests where you can actually run all these tests together. There's a needle, and you put the needle in your bacteria, your one species, and then put the needle in here, and then you can run 12 tests at one time. Any question about any of that? All right, the first biochemical test we're going to talk about is the indole test. Some organisms can produce the enzyme tryptophanase that can convert the amino acid tryptophan to the products indole and pyruvic acid. After overnight incubation, Kovacs reagent is added, which will react with indole to produce a red color at the surface of the tube. E. coli tests positive for indole production, and something like Klebsiella pneumoniae and Streptococcus, uh, Streptococcus, I'm forgetting its name. What the heck is that called? Well, it doesn't matter which one, but Streptococcus cephalophyticus, or what the heck is the number one? Oh, I'm not saying it right. Staphylococcus cephalophyticus is negative for indole production. Normally, when Kovacs reagent is added to indole, it looks cherry red, which is a reddish color or a pinkish red color. This picture is a little bit off in color, or maybe it's using a filter, and it's normally not purple like this. But positive is a red color, and the negative is a yellowish color. Remember, tryptophan, an amino acid, which can be found in most proteins, in the presence of tryptophanase, make indole with Kovacs reagent makes that cherry red or red color for the indole test. All right, let's move on to the next test, the methyl red and Voges Percure test. These are two tests. I'm going to call it the MR test and the VP test just for short, or two tests where you normally inoculate one tube, grow the tube up, incubate it, and then split the tube into two parts, one part for the MR test and one part for the VP test. Both tests, the MR and the VP test, are used to detect the end products formed from glycolysis. Acid products will show up in the MR test, and a neutral product will show up in the VP test. So we have glucose going through glycolysis. If it makes an acid end product, you can determine that from the MR test. If it makes a neutral product, you can determine that from the VP test. You inoculate the MR-VP tube for 2 to 5 days. The MR test is best to grow for 5 days, but the VP test can be grown for as short as 2 days. After incubating the tube, you then split it into two, using one portion for the MR test and the other portion for the VP test. MR, that's methyl red, is added to the portion of the tube for the MR test. If the bacteria produce acid products, the pH indicator, methyl red, will remain red, and that's a positive result in the MR test. On the other hand, if the methyl red is added, and it's always red in color when it's added, if it quickly changes yellow, yellow, that's MR negative. So MR positive is red. MR negative is yellow. The VP test, you have to add the Barrett's reagent A and Barrett's reagent B, and then add oxygen to the tube by shaking the tube, and then letting the tube sit for about 60 minutes. Acetone will then react with Kovacs reagent, acetone being a neutral product, and in the Kovacs reagent, which is always on top of the solution, will be reddish. If it's VP negative, it will once again be yellowish. So for all three of these tests, the indole test, the MR test, and the VP test, if the test is positive, the tube will be reddish. If the test is negative, the tube will be yellowish. So that should be an easy way to remember these three tests. Let's move on to the citrate utilization test. This test is used to determine whether the bacteria have the ability to utilize citrate. In this tube, citrate is the only carbon source and the only energy source. If the bacteria can utilize citrate, remember citrate is a molecule of importance in the citric acid cycle. If the bacteria can utilize citrate, it'll grow on the tube and turn the tube bluish. If the bacteria cannot utilize citrate, it will not grow because it has no carbon source, has no energy source, and the tube will remain greenish. So citrate positive is bluish, citrate negative is greenish. The tube turns blue from an increase in the pH. So this is alkaline, and this is, well, not alkaline, either neutral or slightly acidic. I think it's neutral, but I'm not positive. All right, any questions about the citrate test? Give you any examples on that one? I did not. I think E. coli is MR positive, and that's about the only one I remember off the top of my head. In the citrate utilization test, E. coli and Shigella cannot metabolize citrate, so they'll be citrate negative, and Citrobacter, forgot its name. One of the Citrobacters is positive. What's another one that's positive? I don't remember. I have to look it up, but I think Micrococcus luteus is citrate negative, but Micrococcus varians is citrate positive. Is Micrococcus varians? Yeah. All right, let's move on to the PR carbohydrate metabolism. The PR carbohydrate metabolism test can be called the glucose fermentation test, the sucrose fermentation test, the lactose fermentation test, because in the carbohydrate fermentation test, you have one sugar. And if the bacteria can ferment that sugar, it'll produce acid end products, changing the media from reddish or pinkish color to yellowish, meaning acidic. If fermentation can happen, it can happen without gas being produced, in which case the normal inverted Durham tube will be full with media. And if gas is produced, then you'll get a bubble in the inverted Durham tube. So how you read this tube or explain the results is this would be a carbohydrate ferment test negative, like glucose fermentation negative. This would be glucose fermentation A for acid, meaning it's yellow. And this would be glucose fermentation AG for acid and gas production. Any questions about the carbohydrate fermentation test? The inverted Durham tube is there to catch bubbles being produced by the fermentation. You only see bubbles being produced if fermentation occurs. All right, let's talk about the motility test. Bacteria that possess a flagella are motile. Bacteria that lack a flagella are non-motile. In the motility test medium, we have the medium semi-solid, meaning it's very low percentage agar, and that way the bacteria can actually swim through the agar if they are motile. If they are not motile, and then what you do is you inoculate a needle and then stab it into your motility medium and pull out the needle straight. And if the bacteria only grow in the stab, then the bacteria are non-motile. If the bacteria grow everywhere in the tube, as shown here, then the bacteria are very motile. Let me blow that up a little. So the reason why that's cloudy everywhere is because the bacteria are swimming and they're going everywhere in the tube. So that bacteria is very motile. If the bacteria are slightly motile, they will not look like that. They will look like, here I go. Yep. If the bacteria are slightly motile, they'll be flared. So besides growing in the stab, make the media about here. The bacteria will be growing flared away from the stab mark. We'll make that one similar, and this one different. And that one a little bigger, but similar. So that's what the flare will look like. Any question about any of that? And this is a slightly motile organism. It can swim, but it doesn't swim very fast. And so the bacteria is flared away from the stab. Oops, too far. Shigella and Klebsiella are non-motile. E. coli and Citrobacter, can't remember the name of that one again. Don't remember. One of the species of Citrobacter are motile, and both of them are very motile, so they will be cloudy all over the tube. If you look there, this area there looks clear. You can actually partially see through it, and that one is opaque, so that is cloudy, meaning very motile bacteria. All right, the hydrogen sulfide production test is a test to look for the production of hydrogen sulfide, which is a gas. And we have something that reacts with the hydrogen sulfide and then forms a black precipitate. And we have three different medias to test for hydrogen sulfide production. There's the triple sugar iron, or TSI tube right here. And then there's the Sims test right here. And then there's the HE plates right here. And all of them, if the bacteria are black, that means that they are making hydrogen sulfide. So this tube This species is making hydrogen sulfide. These are not. The others. This tube is making hydrogen sulfide. These tubes and that one are not. So you have three tests you can run to determine if hydrogen sulfide is being produced. The SIMS test, the TSI or triple sugar iron test, and then the hectone enteric or HE plates. Let's talk a little bit about the triple sugar iron test, TSI. It has three sugars, glucose, lactose, and sucrose, but the glucose is present in 1/10 the amounts of the lactose and sucrose. It also has the phenol red indicator, which will be yellow when it is acidic, and reddish when it's neutral, and pinkish when it is basic, or red pinkish when it's basic. It also contains sodium thiosulfate and ferrous sulfate, which allow TSI to detect hydrogen sulfide production. What you do is you get a needle and you put bacteria on the tip of your needle. You then inoculate the slant going down about 2/3 into the butt. The butt is actually from this region of the tube to this region, the lowest portion of the slant. And you want to go, this person has only gone down about halfway, at least so far. You want to go about 2/3 of the way down. You don't want to go further because if the needle touches the glass of the tube, it could damage the needle. It certainly could bend it, and you need the needle to be fairly straight. You then pull out the needle, and the purpose of growing the bacteria and the TSI's slant test in the butt is to look for the growth of the bacteria under anaerobic conditions. That's why we inoculate the butt. And then you take the needle out and once again put bacteria on the tip of the needle and then inoculate the slant with the bacteria in an S-like motion. Then you look at the growth in the slant, which is growing aerobically, and in the butt, which is growing anaerobically, or at least partially anaerobically. Then you incubate the tube and record the color of the slant over the color of the bud. You record other things too, but that's the main one. And unfortunately, in this test, it's very complicated. So we'll talk about tube one, that's a negative control. No sugars are fermented because there's no yellow. No gas being produced because there's no bubble, no cracking of the agar. No hydrogen sulfide being produced, but it is not blackish. Then I'm going to move over to tube 4 because it's the easiest to describe. It is yellow in both the slant and in the butt. Because it's yellow in both the slant and the butt, then we know that glucose is being fermented as well as one other sugar or both other sugars. So either lactose and or sucrose are also fermented. Gas is produced in this because we have a bubble here, as well as the breaking or cracking of the agar up there. No hydrogen sulfide is being produced because there's no black in the tube. Any questions about tube 4? All right, the next easiest, I guess, is tube 3. We have a slant, which is yellow, and then, excuse me, a butt, which is yellow, and then a slant, which is pinkish or reddish. Because the slant is yellow, because the slant is pinkish or reddish and the butt is yellow, we know that only glucose is being fermented. The reason why the slant is pinkish is because all of the glucose has been used up when the bacteria are growing aerobically. But not all of the glucose is used up in the slant because anaerobically, the bacteria use glucose slower than they do aerobically. And that's why the slant is yellow and the butt is yellow and the slant is pinkish or reddish. If you were to look at this test earlier, you would have seen the slant yellow, meaning initially the bacteria use the glucose, ferment it, produce acid products, turning the slant yellow. But in the time span of this test, the glucose gets used up, and then the bacteria, rather than die, start utilizing the protein, and then they make ammonia as their waste product. And ammonia is basic if you don't know, and that will change the color of the slant from yellow to pinkish or reddish. So if only glucose is fermented, the slant will be pinkish or reddish because the glucose got used up. But in the butt, the glucose is fermented slower, so the butt remains yellow from glucose being fermented. Any question about that? And the two other sugars are not utilized because if they were utilized, they would still be making acid products in the slant, and then it would be closer to tube 4. Anyways, that's tube 3. Let me state there's no gas being produced. There's no bubble in the tube, no breaking of the agar, so no gas is being produced. There's no blackening of the agar, so it too means there's no hydrogen sulfide being produced. And that's tube 3. For tube 4, we notice that the slant is red and that the butt is black. Whenever the butt is black, that means hydrogen sulfide has been produced or is being produced. And hydrogen sulfide is only produced when fermentation happens. So we can't see it, but we know that the butt is yellow. And you can kind of guess it from this because this should be that minus the black. And so the bottom of the tube is assumed to be yellow, and it is, but you can't see it because the black hides it. And only glucose is fermented because the slant is pinkish. Any question about tube 2? One other thing about tube 2, no gas is being produced because there's no bubble in the tube. There's no breaking or cleaving of the agar. And that's it for TSI. Any questions about TSI? Sorry, it's a complex test. Just do your best trying to remember. I think on the final, there's one question about TSI. Let's move on to the SIMS test, which stands for Sulfide Endomotility Test. What you do is you inoculate the SIMS tube in the center with an inoculating needle. If I blow this up, actually I got it blown up. Right there, you can see the stab mark there, bacteria growing on the stab. There too, there it's so more difficult to see. I'll talk about that in a minute. And there. In tube 1, you can't see the stab, but it's there because the bacteria are everywhere. Excuse me, this bacteria is very motile, and that's why it's black everywhere. So, of these tubes, once again, if the tube is black, hydrogen sulfide is being produced, and that's making the tube black. Once again, hydrogen sulfide is made under anaerobic respiration, is made under anaerobic conditions. So usually only the bottom of the tube will show blackening. And let me see right here. That's why the bottom of the tube is black and the the slant is not black. There's only black usually happens anaerobically. Doesn't happen aerobically. And then you add Kovacs reagent to the top of the tube. That's that yellow there. So that's Kovacs negative, meaning indole is not being produced. These are all pink. And that's because indole is being produced. That one is yellow or yellow-white. And so Kovacs reagent is not being made in tube 5. Any questions about the hydrogen sulfide production test and the indole test for the SIMS tube? And then M of SEM stands for motility. We're also doing a motility test here. What you're doing is you're stab inoculating the tube. And if the bacteria grow out of the stab, then the bacteria are motile. But if they only grow in the stab, then the bacteria are not motile. So tube 2 is not motile. Tube 3, it's hard to see because it's kind of cloudy in this region, but that's just the photograph. The bacteria are only growing in the stab mark, so the bacteria are non-motile in tube 3, and I'm sure that's in there. Yeah, motility negative. Tube 5 is also motility negative because bacteria are only growing in the stab. Tube 4, on the other hand, has bacteria flaring, a very large flare right there, out of the stab. So this bacteria is somewhat motile. It's not very motile, but it's somewhat motile. A little more than slightly motile, but somewhat motile. And then in tube 1, it's cloudy everywhere. So the bacteria swam everywhere. It's also black everywhere, and the bacteria swam everywhere, allowing the black to be everywhere in the tube, except obviously where the Kovacs reagent is. And so tube 1 is positive for motility. Excuse, yeah, tube 1, as well as tube 4. Tube 1 being highly motile, tube 4 being moderately motile. Meaning that's a pretty big flare. That's why I'm calling it moderate motile. All right, when we're growing things on McConkey's agar, we are selecting for gram negatives and selecting against gram positives. What's doing the selection is bile salts, which are in people's intestines, and gram negatives can usually tolerate bile salts, but gram positives do not, and they don't like growing around bile salts, as well as the fact that we have crystal violet in the media, why the media looks kind of purplish there. Crystal violet gets in the gram-positive cell wall, gets stuck in the cell wall, and then prevents the cells from growing. So if there's growth on the McConkey's plate, it's likely to be gram-negative. If there's no growth, it's likely to be gram-positive. It applies to 95% or more of bacteria, but not all bacteria. All right, let's finish up the McConkey's plate. If the colonies or the growth is pink, as well as pink around the bacteria, then the lactose is fermented, and the pink color happens because of acid products from the fermentation of lactose. If the bacteria cannot ferment the lactose, we call them non-lactose fermenting, then the bacteria will be their normal color. For this, the bacteria are either white or off-white, and there's no pink in between the bacteria. Any questions about McConkey's plate? For hectone and enteric agar, once again, if the bacteria can grow, it's likely to be gram-negative, because gram-positives do not like growing on HE plates, so they're selected against. If the bacteria can grow on the plate, and it forms a salmon color that's yellow-orange color, then the bacteria can ferment the sugar lactose. If the bacteria cannot ferment the sugar lactose and they do not produce hydrogen sulfide, then the bacteria will be their normal color. And in this case, the colonies are white, and that's clearly not salmon colored. So these are non-lactose fermenting gram-negative bacteria. If, on the other hand, the bacteria can produce hydrogen sulfide, then the colonies will appear black, regardless of whether the bacteria can ferment lactose or does not ferment lactose, the colonies will be black. How you can determine whether this colony is non-lactose fermenting with hydrogen sulfide production, or if it's lactose fermenting with hydrogen sulfide production, is you can look around the colony. Oops, too far. And there you can see a colony and another colony. And then you can see this orangish, reddish area around the colonies. This is not where the colony is. This is changing the color of the medium. And if you see that, and the colony is black, so instead of being yellow, orange, or salmon colored, it'll be black there. But you see the orange color in the media, then you know the bacteria is fermenting the sugar lactose, as well as producing hydrogen sulfide. On the other hand, if you don't see the media turning orangish or yellow orangish color around the colony, and the colony is black, then the bacteria is non-lactose fermenting, and it is producing hydrogen sulfide for why the colony is black, and then why the media around the colony is not salmon colored. Any question about HE and McConkey's plates? Shrink that down a bit. All right, you're not going to be physically performing any of these biochemical tests because everything we do will be done online. However, you will be held responsible for knowing how to perform and interpret these tests. We have some video clips for you to watch. First, on carbohydrate fermentation test. Second, video on the SIMS test. A third video on MRVP test. Remember, this is two tubes that get inoculated into one tube. You grow it up, and then you split the tube in half, using half for the MR test and half for the VP test. You have a little video for you to watch on the TSI test. And then lastly, a little video for you to watch on growing your species on McConkey's auger. The MicroBuzz website did not work the last I checked. However, all the information you need for Lab 9 is actually either in this lab module, most of it is in this lab module, but there might be something in the question that comes from a previous lab. But I will tell you that all the questions can be answered just by looking up the answer in the lab modules if you do not know. Any question about any of that? All right, let's go ahead. MicroBuzz can also tell you about the different tests. So if you wanted to read more material about the test, you can read it on MicroBuzz. All right, let's go to the worksheet and fill in the first table. Wrong one. Right there. Well, can't get to it. There it is. So the first example has been filled in for you, but in question 1, fill in the table. For the oxidase test, tell me what happens in the positive result. And I filled it in for you. The spotter cells turn blue, and then give me the negative result. Do the same for indole. For the positive result, give me the color. If the indole test is positive, give me the color if the indole test is negative. For the MR test, to give me the color if the MR test is positive, give me the color if the MR test is negative. Do that for VP test, positive result here, negative result there. The citrate test, give me the color for the citrate test. If the result is positive, give me the color for the citrate test if the results are negative. Moving on to carbohydrate fermentation, we split the carbohydrate fermentation into whether the sugar is fermented, like whether glucose is fermented, and then if gas is produced. So give me the color of the tube if the sugar is fermented. Let's say glucose. If glucose is fermented, give me the color of the tube. If glucose is not fermented, give me the color of the tube. If gas is produced, then tell me what you see in the inverted Durham tube. And if the gas is not produced, meaning the negative gas produced, then tell me what you see in the inverted Durham tube. Move on to TSI. We're looking at glucose fermentation, that one sugar in TSI. For the results, you need to give me the color of the tube in the slant over the color of the tube in the butt. If you only give me one color, the answer is half wrong. So for glucose fermentation, you need to give me the color of the slant over the color of the butt in both the positive and in the negative. Color of the slant or the color of the butt. And then if lactose and or sucrose is fermented, tell me the color of the slant over the color of the butt. And then tell me the color of the slant over the color of the butt if lactose and or sucrose are not fermented. For TSI test, if hydrogen sulfide is being produced, tell me the color of the tube for the positive result and the color of the tube for the negative result. If gas is produced in TSI, tell me what you see in the positive result and what you do not see in the negative result. Any questions about TSI test? And all of these tests are ones that are in the lab module, so there's nothing really you need to look up. Maybe you'll need to look up oxidase, but all the others are in this lab module. And you can find oxidase in a previous lab. All right, let's move on to the SIMS test. Tell me the color you see if sulfide is produced, meaning hydrogen sulfide gas is produced. Tell me the color of the tube if hydrogen sulfide is not produced. If indole is produced, tell me the color of the Kovacs reagent on top if indole is produced. And then tell me the top of the tube, the color of the top of the tube where the Kovacs reagent is for a sulfide production negative test tube, meaning the negative case. And SIMS, tell me what you see for motility in the positive case, and tell me what you see for motility in the negative case. Any question about that? And then for selective and differential media, meaning McConkey's and HE plates. Tell me what color you see for MAC and HE if lactose is fermented, and tell me the color you see for MAC and HE if lactose is not fermented, meaning the negative case. Any questions about this table? Not hearing anything, let's move on to the section 2. Complete the following sentences. For 2A, which reagent must be added to detect the presence of indole? Put that in 2A1 here. For 2A2, if indole is present, the test will show what color. Put that here. For 2B1, methyl red is a pH indicator that remains what color under acidic conditions. And the methyl red pH indicator is used for what test? For 2B2. For 2C1, the what test uses Barrett's reagent A and Barrett's reagent B to detect the presence of acetone, a neutral product formed from glucose metabolism. So name the test in 2C1. And then in 2C2, if acetone is present, the test will show what color? Answer that there. For 2D1, Simmons citrate uses the indicator, pH indicator, and give me the name of the pH indicator here. And this indicator turns a blue color under which acidic or basic conditions. You can bold your answer, you can underline your answer, you can highlight your answer, or you can write your answer right here. Any question about any of that? For 2E1, what is the pH indicator used to detect PR carbohydrate fermentation? And then this pH indicator turns yellow under bold, right, or underline acidic or basic conditions. So either underline that, highlight that, or you can just write it there. Any questions about 2E? If not, let's move on to 2F. Bacteria that possess this structure test positive for motility. Name me a cell structure. Do not go in and name me like a test or something like that. Name me a cell structure that makes the bacteria motile. For 2G, give me the names of 3 organisms that produce hydrogen sulfide, and you were given 3 in the lab module. You can also look online. If an organism ferments glucose, but not lactose or sucrose, the TSI slant will be what color, and the deep will be what color? So TH2H1, give me the color of the slant. For 2H2, give me the color of the bud. For 2I, state the 3 tests for which SIMS media is used. You don't have to put it in exact order, but you must get the initials or the name of the test. For 2J1, MAC and HE plates are both what? In that they inhibit the growth of either gram-positive or gram-negative. So what's a plate that inhibits the growth of either gram-negative or gram-negatives? What's that called? And then for 2J2, write, bold, or underline gram-positive or gram-negative, meaning MAC and HE plates do not grow either gram-positive or gram-negative. You can write the answer there, or you can highlight or underline one of the answers here. Any question about 2J1 or 2J2? There's also a 2J3, and these plates, meaning MAC and HE, are also what? and that they separate various bacteria based on visual differences from sugar fermentation. Any question about that? All right. So in the worksheet, I have the case study key. Here, use this key only for this lab. Do not use it for your unknown project. Do not use it for any other lab. Only for this lab. And then I have a table here of case study 1 and case study 2. Fill in the test results for the different tests for case study 1 and the other case studies here. So put in the results here. And then when you get the results, like case study 1, if it's oxidase positive, well, then it has to be this one because that's the only oxidase positive one. So you don't even have to go further. But if it's oxidase negative, it's one of these. And then if it ferments glucose, it's actually all of them here. If it ferments lactose, it's narrowing it down to these 4. And TSI, if it's A over A, it narrows it down once again to these 4. If it is hydrogen, produces hydrogen sulfide gas, then it is either Citrobacter furundi or Salmonella. And that's the Citrobacter I was trying to remember, Citrobacter furundi. So if it's positive for hydrogen sulfide, then it has to be Citrobacter furundi or Salmonella. And look, we can already cross out Salmonella because we know that the TSI is A over A, as well as the fact that it ferments the sugar lactose. And that means we've excluded Salmonella. And that's how you go through a case study, by the way. All right, let's briefly look at one of the case studies. Oh, I'm in the wrong place, sorry. The case studies are in the bottom of the lab module. For case study 1, we have a prison inmate brought into the ER room with an open wound, and this prisoner is known for inflicting. A routine culture is ordered, and the appropriate transport media is used for specimen collection and inoculation of the prescribed media. Below are the results of the routine culture. Here we're looking at a blood agar plate, and this is the bacteria grown on the blood agar, and you can see the hemolytic pattern. We then grew the bacteria on McConkey's agar, and here it is here, and you can see from the color that something's going on McConkey's agar. And there's the gram stain. You need to blow that up in order to see it. I've already got this 140, and that's not big enough. Let's go to 200. You might even have to go a little higher. Aha, I can see it now. Let's blow it up just a little further. You can see clearly. Right here, it's hard to see, and there, hard to see what the cells are. I can't see them there, but that'll be hard for you guys. But right there, and right there, and there, it's pretty easy to see what they are. And you should be able to tell the gram stain as well as the shape of the bacteria. You should not determine the cellular growth arrangement from the gram stain. We then do some further tests, and we find out that the bacteria is catalase positive and oxidase negative. And then all of these you put in the worksheet. Let's see if I can get that to come up now. There it went. So case study 1, state the oxidase test. fermentation of glucose, fermentation of lactose. Not about TSI yet. H2S production, you can get that one. I haven't talked about the MR and the VP test, but we're starting to fill in the table here. And then let's go back to the lab module and look at the other tests. So the test for day 2 is, this is the SIMS tube, running 3 tests at once. This is the MR test. This is the VP test. That's the cinnamon citrate test or the citrate test. And this is the TSI test. So write your results in the table, in the worksheet here, and then use the table and the results with the key up here for identifying the species. For example, if it's oxidase negative, it's all of these species. If it can ferment glucose, it's also all of these species. And then if it ferments lactose, it's one of these 4. If it's TSI A over A, it's also one of these 4. But if it's H2S production, it's only this one, because that one we've already excluded from the fermenting lactose. Everyone see how to do this? That's how you do the case studies. And then write your organisms in the worksheet. For case study unknown 1, write the species if the species is given. If only the genus is given, like Salmonella and Shigella, as well as Pseudomonas, you only need to write the genus. But you should write the answer. And if you can give the exact species, give the exact species. Although all of these are in different genuses. So write your answer in here and then submit your lab before this Saturday, 11:59 p.m. this Saturday, because that's when this lab is due. Any questions? All right, go ahead and get started on the lab. If you have any questions, I'll